Hello everyone, and welcome to your AP Biology Midterm Review Cram Session video. If you're watching this video, you probably have a midterm to study for, so let's get started. I'm going to be doing a really fast content review of some of the hot topics that you might have encountered throughout your year in AP Biology so far. I'm going to be covering units 1 through 5 in the AP Biology curriculum, but your teacher might have taught these topics in a different order than they're going to be presented in the video. Whatever you're studying for, there's lots of other resources on this channel to help you prepare for your exams and do the best that you can absolutely do. And be sure to subscribe if you don't want to miss out on any of the resources on this channel that'll help you prepare for the AP Biology exam at the end of the year. These 6 elements are really important in biology. CHNOPS is an easy way to remember those main elements: carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. But just saying that silly word will help you remember these if you forget. But there's other elements as well that'll come up in biology. Sodium and potassium, for example, have really important roles in things like nerve signaling, biological processes. Let's go back to our atoms really quick. So remember that protons and neutrons are in the nucleus, and then electrons have different shells which they occupy surrounding the nucleus, and each shell can have a certain number of electrons within it. So this is carbon, and carbon has a full first shell here, but its second shell is incomplete, and so that means it can form up to 4 covalent bonds with other atoms. Really quick before we talk about bonds, isotopes are different versions of different elements. So carbon-12 might be the common one that we just saw, but carbon-14 is going to have a different number of neutrons. So these are different versions of elements with different neutrons in them, and that's going to influence the atomic mass. So the atomic mass will change in a different isotope. Some isotopes are radioactive, like carbon-14. They're going to decay over time, and we can use the half-life of these elements to determine the age of things, like fossils. Like we can use carbon-14 dating to determine the age of this mammoth here. So chemical bonds are formed in different ways, either through the sharing or transfer of electrons between atoms. The 2 main types are covalent bonds and ionic bonds, shown here. So this is the structural diagram of methane here, with the carbon in the center forming 4 covalent bonds with hydrogen around it. And then ionic bonds involve the transfer of electrons and really are based on electrical attraction. And remember, ions, like we saw here in sodium and potassium, these are atoms that have gained or lost, in this case, lost electrons, resulting in a positive charge. These are cations here, or a negative charge for anions. Now, certain molecules or groups of atoms that are bonded together can have different properties, like their polarity. So some can be polar, which sometimes are called unbalanced or have an unbalanced charge, and then nonpolar, which have more of a symmetrical balance to them, a symmetrical charge. And this just means that the electrons in those atoms are drawn to one nucleus more than another. And so with water, with our large oxygen and then 2 small hydrogens, we have an imbalanced charge. So we have a more negative side and then more positive side of this molecule. And so water is a polar molecule, and it plays a really important role in biological systems due to its unique properties. Now, hydrogen bonds are really unique types of connections between molecules, and they're relatively weak compared to ionic and covalent bonds, but they're crucial for maintaining the structure of certain molecules like DNA. So in between all of our bases in DNA, we have different numbers of hydrogen bonds. And then, of course, water can connect to other water molecules through hydrogen bonds as well. But they are weaker in comparison to ionic and covalent bonds because they involve just partial charges, but they are really crucial in lots of different biological molecules, DNA here, the structure of different proteins, which we'll see later on. So pH, really quick, another chemistry concept that's important in biology is a measure of how acidic or basic something is. Acids have a pH that range from 0 to 6. Bases have a pH that range from 8 to 14, and substances with a neutral pH are right at 7. So that's like pure water, blood. But just remember that lower values on the scale are going to be more acidic, higher values are going to be more basic. Buffers sometimes get involved, and they're used to maintain homeostasis, which is the internal balance of an organism. In different organisms, 70 to 95% of their structures, of their body structures, is actually made up of water. And in humans, water's unique properties contributes to homeostasis and our survival as well. Water moves in and out of membranes through osmosis, moving from a high concentration of water to a lower concentration of water. In humans, water is a thermal regulator. It helps us regulate our temperature. It helps, obviously, transport materials. Our blood is composed of water, but plants too have plant sap, which is composed of water. It cycles through us. We eliminate extra water through our kidneys. And in our bodies, the liquid water is the most important, but it can also exist exist as water vapor and, of course, as solid ice. So let's talk about those properties. Water is a polar molecule, which means it has an uneven distribution of charge. So one side is more negative and one side is more positive. The oxygen atom is more electronegative than the hydrogen atom, so it pulls electrons more towards itself, which gives that side a partial negative charge, and the hydrogens have then a partial positive charge. And because of this positive and negative side, this leads to hydrogen bonds, or this attraction that happens between water molecules. So remember, it is a solvent. It can dissolve a lot of different things, but because of its polarity, it can stick to itself really well. That's cohesion. It can stick to other substances. That's adhesion. It forms really strong attractions to itself. When there's a layer of water molecules on the surface, it's more attracted to itself than the air above, so that contributes to surface tension. It also has a high specific heat capacity, which means it resists changes to temperature, which means it can absorb and store a lot of heat before its temperature rises, and that is crucial for temperature regulation in organisms and helps us maintain a stable internal environment. It also has a high heat of vaporization, which means it takes a lot of energy to convert from liquid to gas. Add a little bit more vocabulary for review. Again, polar, meaning water has a slightly positive and a slightly negative end. It'll work well with other polar molecules, and non-polar substances will tend to avoid water. So molecules that are water-loving or that interact and are soluble in water are often called hydrophilic, and molecules that are water-fearing, hydrophobic, are insoluble in water. So we think about the structure of our phospholipid bilayer in our cell membranes. Those hydrophilic phosphate heads are going to be more attracted to water, and the hydrophobic tails are going to avoid water, and so they'll situate themselves in the center of that membrane, which you can see right here. Now let's touch briefly on molarity. This is more of a chemistry concept, but we'll touch on it in laboratory settings in biology. Molarity is a measure of the concentration of solute, the thing that's being dissolved, in a solution. It's expressed as number of moles of solute per liter of solution, no moles per liter. So usually we'll see it in the context of a lab when we have a particular solution. We'll see the molarity of a specific chemical or molecule in a biological sample. It's good to recognize that something with higher molarity has a higher concentration of particles within it, whatever that substance may be. So a 10 molar solution would be strongest, 1 molar would be relatively intermediate in this situation, and 0.1 molar would be the weakest in this particular spectrum of molarity. Molarity can often be swapped with molar concentration, but you may often see this in the lab if you're asked to set up a solution or dilute something. And if you need to prepare a solution of a particular concentration, you would use molarity to calculate the amount of solute needed to achieve that concentration in a given volume amount. I'm going to go through the monomers of each of our main categories of organic compounds or macromolecules, talk a little bit about their function, and then some indicators which you might use in the lab or you might have used in the lab to identify each one. So nucleic acids are made of nucleotides, that's our monomer, our single unit, to build our larger things like DNA and RNA. They consist of a phosphate, a sugar, and a base, and then many of them link together to carry genetic information in organisms. Proteins, which DNA code for, DNA actually carries the instructions to build proteins, are made of amino acids. Proteins do all the jobs within cells and living things. They transport materials, they provide structure, they send signals, they receive signals, and they are also enzymes which help catalyze or get chemical reactions going. So they do all the jobs of the cell, and all of our genetic traits are based on proteins because that's what our DNA codes for, is proteins. Now, these amino acids have a very specific structure, which we'll talk about in a moment, but they have one really important piece called an R group that is on one side of each amino acid, and that piece varies from amino acid to amino acid. So different characteristics of that R group are going to give the amino acid different properties, which then changes how the amino acids will fold and link up with each other. So we get these long polypeptides or these long chain of amino acids, and every amino acid influences how the protein is going to fold and its final structure and then its function. Carbohydrates, like I said before, are made of monosaccharides like glucose, which is a simple sugar, and then larger polysaccharides are built from those monomers all put together. So many glucoses put together will give us cellulose, which provides structure like in the cell walls of living things. But on its own, carbohydrates are responsible for energy. So we use glucose in cellular respiration and in fermentation to provide energy to living things. So that's really important as far as function goes. Lipids are going to be made of fatty acids, triglycerides, but one example that we see in every living thing are our phospholipids, which are what the membrane is composed of, the cell membrane. One thing these things all have in common, despite their different functions and structures, is they all contain carbon. That's why they're called organic compounds sometimes. And they also contain hydrogen and oxygen. Now, they have other elements present in them as well, but they all contain carbon, hydrogen, and oxygen. Let's take a closer look at proteins really quick before we move on. Remember, proteins are long chains of amino acids, and each amino acid is one monomer, one subunit, and it has that carbon in the center with the hydrogen attached to the top. And remember, carbon can make 4 bonds. One of those will be to the hydrogen, one of those will be to what we call an amino group, so that N and 2 H's, and one of them will be to a carboxyl group. Now, the protein will grow by attaching more amino acids on the side of that carboxyl end. But then the side chain, that R, isn't just an R, that's not an element, that is just a placeholder for whatever other molecules are going to attach to the amino acid and give it its properties, like maybe being hydrophilic or hydrophobic or ionic and more acidic or basic. And so every amino acid has different characteristics that are based on this side chain or this R group. DNA, of course, is super important too. If we go back to nucleic acids as one of our categories, and just like water, in the center, it's connected by hydrogen bonds, which can easily break and then come back together when we need to replicate the DNA or undergo protein synthesis. And you can see all of these nucleotides built together to form the structure of our DNA double helix. So different cells will have different organelles depending on the functions that they serve. Prokaryotic organisms are going to have different organelles than eukaryotic organisms. And remember, prokaryotes are organisms that lack a nucleus and membrane-bound organelle, so they're going to be relatively simple compared to our eukaryotic organisms. They're going to be pretty small, and in general, they will have fewer components. Our eukaryotic organisms, including plants, animals, protists, fungi, these will all have cells with membrane-bound or organelles that are surrounded by a membrane. And we can compare different types of cells like plant and animal cells, both eukaryotes, and see, for example, that a plant cell has a chloroplast and an animal cell does not. Now, these are the organelles that you really should be able to know, recognize, and understand how their functions work for AP Biology. There's a lot of other organelles out there and some major ones that are not on this list, but not on the exam. These are the ones that might show up and you might be tested on. Out of this list, the most important ones you really need to know and recognize are cell membranes, mitochondria, and chloroplasts. A few other organelles I wanted to mention include the endoplasmic reticulum. We both have the smooth ER and the rough ER. The rough ER contains ribosomes. And this organelle is a network of tubules and sacs that helps transport proteins and other materials within the cell. This right here could be a lysosome, maybe a peroxisome, but it's a lysosome that is a membrane-bound sac that has enzymes that helps break down waste products and other damaged organelles, kind of like a garbage disposal of the cell. And then, of course, the Golgi apparatus or Golgi body that is responsible for packaging and shipping materials within the cell or out of the cell. So overall, cells are comprised of different defined compartments. In eukaryotic cells, we know these as organelles. Each compartment has a specific function, and generally it is a eukaryotic feature, but there is some compartmentalization in prokaryotic cells as well. But as we know, eukaryotic cells have well-defined compartments. Each part has a specialty for a particular function, and a lot of times when we focus on different processes within the cell, we focus on where they occur, whether that is within the plasma membrane, within the cytosol, or within membrane-bound organelles like our Golgi apparatus or our nucleus or our mitochondria. Let's take a quick look at prokaryotic organisms. So prokaryotic cells are much smaller than eukaryotic cells. And why in general are cells so small? We know that as a cell increases in size, its surface area to volume ratio decreases. When we have not enough surface area to support a cell's increasing volume, a cell is either going to have to divide or it's going to die. Because we have a decreased rate of chemical exchange when the surface area to volume ratio is low, this results in death of the cell because the processes that the cell needs to perform can't perform at the rate that they need to. Again, if the surface area to volume ratio is too small, meaning we have not enough surface area to support the volume of the cell is not going to be able to support all of the chemical functions it needs to do. Cells need to stay a certain size in order to support all of these functions, so we want a greater surface area to volume ratio wherever we can. We'll see this theme come up again and again in different cellular structures, different tissues, as well as organs. Humans, for example, have a brain that has a good example of surface area increase with all of its folds and gyri and sulci. So why are organelles important and why do we even have them? Again, there are 4 compartmentalizations. Each particular compartment within the cell has a specific function it needs to perform. It provides a favorable local environment for metabolic reactions. Certain metabolic reactions have very low or very high pHs compared to the rest of the environment of the cell, which means certain enzymes within those compartments wouldn't function if they were combined in the rest of the environment of the cell. In order for the enzymes and other proteins to do the jobs that they need to do, they need to be in the environment that works for them. So the compartments can lead us to that. Cells have these membranes that allow them to establish and maintain our internal environments that are different from the external environments. We minimize competing interactions, and we increase surface area where reactions can occur. For example, along the cristae, the folds of the mitochondria. And again, it could also protect different parts of the cell from potentially damaging metabolic reactions, when the pH or byproducts of reaction end up being dangerous for the rest of the cell. So how did we get organelles? The theory of endosymbiosis is one of the most popular ones for how membrane-bound organelles evolved once from free-living prokaryotic cells. So this idea that chloroplasts came from free cyanobacteria within an environment that eventually were engulfed through phagocytosis and adopted a symbiotic relationship with larger cells. Later on, these became parts of the cell that were synthesized as the cell replicated as well. But that engulfing and then that symbiotic relationship we think was the origin of certain organelles such as the chloroplast and the mitochondria. We have evidence for this due to the fact that many of these organelles have a double membrane, so our phospholipid bilayer, just like the one that surrounds the cell. And so based on their size and the membrane structure, it's very likely that these organelles once were their own type of cell. Mitochondria themselves have their own DNA, another hint that they were once prokaryotic cells themselves. They also support energetic processes, and these internal membranes help with compartmentalization and efficiency within that organelle. Another skill that you'll need to know is how to calculate a ratio. This one is another pretty simple calculation, but you're just comparing one data point to another data point, so you're dividing data point A by data point B. So we would see this maybe in a situation where we're looking at surface area to volume ratio and comparing which cell might be more efficient in exchanging materials or removing waste by diffusion. So if we just look at these two cells, what we want to do is see which one has the greatest surface area to volume ratio, and that'll be the cell that is more efficient. In this case, it's already given the surface area and volume, so you don't have to calculate those separately. But to calculate the rate, we just divide our first value by our second value, value A by value B. So in this case, that's 600 divided by 1,000 to give us 0.6. And then for our cell B, that's 5,400 divided by 27,000 to give us 0.2. In this case, cell A has the greater surface area to volume ratio, and so it is more efficient at exchanging material. But the membrane is really crucial for containing all the important materials of the cell. So as we mentioned in other videos, the cell membrane is a thin layer that surrounds all cells. It's made up of a phospholipid bilayer. So those phospholipids are lipids, and they have phosphate heads, which are hydrophilic, that will interact with water well, and then tails, two lipid tails, which are hydrophobic, that tend to avoid water. They are water avoiding or water fearing, according to the name. The way the bilayer, the phospholipid bilayer, arranges itself, and right here we're just looking at a cross-section of a membrane. Remember, this is all the way around the cell. So if we were like zooming in on one single section of our cell membrane, this is what we're looking at. But the outside of the cell will have the heads facing outward, and then the inner components of the cell will also have those phosphate heads facing inward too. So the tails arrange themselves in the center of that double layer. These pink dots we will come back to. Right now they're just representing different particles that could be outside or inside the cell. Now remember, all cells, whether eukaryotic or prokaryotic, will have a cell membrane, that phospholipid bilayer. Some eukaryotic organisms and some prokaryotic organisms may also have a cell wall that is an extra layer of protection and support that surrounds the cell membrane, but all cells, no matter what they are, even if they do have a cell wall, do have a membrane as well. Now let's dive a little bit deeper into some of the other things that are happening within the cell membrane and what makes it so special. Now it's not just phospholipids, even though that's sometimes what are depicted, and it's not just transport proteins, which you might see in diagrams, and we'll get to those in a moment as well. There's also other integral proteins, peripheral proteins, carbohydrates, cholesterols, and each of these serve a different purpose within the cell membrane. We call it a fluid mosaic because there are lots of different pieces and components. That's the mosaic, and then fluid because it is fluid. There is motion to it, it's flexible, and one of the most important molecules, brain, that is going to help stabilize it but also regulate the fluidity is the cholesterol molecule, which is a lipid as well. And we can see where it's found within that bilayer, again towards the center, since lipids in general are hydrophobic. Another important molecule in the membrane you might hear about are carbohydrates, which primarily we will see on the extracellular side, so on the outside of the cell membrane, and they're used for various purposes, from protecting the cell to signaling to other molecules to serving as identification markers on the cell. A lot of times we'll find them attached to glycoproteins, and there'll be other proteins as well that can be found on the outward facing side of the cell membrane that are going to help for signaling. They're going to help with identification of the cell or help the cell adhere or stick to other cells. There's a lot of different functions that these membrane proteins could have. Now, often when we talk about proteins within the cell membrane, we are talking about transport proteins, which are going to allow certain molecules in and out of the cell. Now, the cell membrane serves as a protective barrier. It helps it communicate with the environment, but it also helps regulate what goes in and what comes out of the cell. And that's where sometimes these transport proteins get involved. So let's take a look at how molecules can get in and out of a cell, starting with passive transport, or transport that doesn't require any additional energy. So first of all, there's simple diffusion, and these are really small molecules that are able to go from high concentrations to low concentrations without any extra energy added, and they don't need a special pathway or a door, quote unquote, to get through the membrane. We'll see diffusion happen with nutrients and gas exchange, so the diffusion of oxygen into the blood. But if we're looking at a diagram, if we see molecules moving from a higher concentration to a lower concentration and there's no protein, no ATP, then we can identify it as simple diffusion. Now there's also facilitated diffusion, which often occurs with larger molecules or molecules that aren't able to interact with the cell membrane because of their charge. And they will also go from a high concentration to a low concentration, high outside the cell, lower inside the cell, or vice versa. And no energy will be needed for this process, but they'll use these either carrier or channel proteins to get inside the cell. Again, both of these are passive transport. Finally, we get to active transport, which does involve ATP or energy. And this is when we are moving from a low concentration to a high concentration. So we're going against the concentration gradient, and molecules are being sent in a way that requires extra energy. And we're going to use an extra transport protein to have this happen. All right, one last type of transport that I wanted to mention for today is osmosis. Now, osmosis, simply put, is the diffusion of water. So it's when water moves across the membrane from a high concentration to a low concentration, and often occurs when there are differences in concentrations of solute, so substances dissolved in solution that can't cross the membrane, but water can. And in general, water will move towards where there is more solute, even though the water itself is going from a high water concentration to a lower water concentration. So let's look at a few examples. If we have a cell and the concentration of particles in and out of the cell is relatively the same, that's called an isotonic solution. Equal on both sides. Water is still going to move in and out, but it will do so at approximately an equal rate, and so we won't have any net gain of water or net loss of water out of the cell. Now, if we have a situation where there's actually a higher concentration of solute, of particles inside the cell, and a lower concentration of solute outside the cell, what's going to happen is we have a hypotonic environment. Think hypo meaning low, so low solute. And if we think of the term solute sucks, we can remember that water is going to move towards where there's a higher solute concentration, but think also about where there's a higher water concentration. Remember, water itself will move from where there's high water concentration to a lower water concentration. So there's a higher water concentration, apparently, on the outside, a lower water concentration comparatively on the inside, so water will move into the cell. Often this may cause the cell to expand or even burst, but that depends on the situation. Now in this environment, we have a higher concentration of particles outside the cell, a lower concentration of particles inside the cell. This is called a hypertonic environment. Think hyper, like excited, like there's a lot, so there's more particles outside the cell. And again, water will move from a higher concentration of water to a lower concentration of water. So in this case, it will move out of the cell. The cell may even shrink. Enzymes are extremely important molecules in biology. They are going to be catalysts that help certain reactions occur. They can help break down certain molecules or put other ones together. They're important in photosynthesis and cellular respiration and digestion. In almost everything that's going on in the cell, enzymes are there and they are key. We'll talk about enzymes in medicine and health. Think about people who are lactose intolerant. It's because they don't have enough lactase enzyme to break down lactose, that milk sugar. In the environment and ecology, we'll continue to talk about enzymes. And remember that enzymes help reactions happen. Now, one key way to recognize an enzyme, even if you've never seen it before, is the letters ASE. Now, not every enzyme ends in the letters ASE, but many do. Even things like Rubisco, their full name is a word that ends in ASE as well. Let's take a closer look at a basic enzymatic reaction. So we have an enzyme, which remember are proteins. We'll get back to that in a minute. So we have this specific configuration of amino acids with one part called an active site where it's going to bind to a substrate or the molecule that is going to undergo the reaction. It's the reactants. This substrate will then attach to the enzyme and make what's called the enzyme substrate complex. The reaction will occur and then the products will be released. The enzyme is unchanged and can complete the reaction again and again. We call enzymes reusable because of this. And if you have enough enzyme to complete the reaction, then it'll keep going and going until there is no more substrate to act on. Now, let's get back to this idea that enzymes are proteins. So remember our four main categories of biological macromolecules: carbohydrates, lipids, proteins, and nucleic acids. Enzymes are in the proteins category. And it's important to remember that because it's important to remember what enzymes are made of, and that is amino acids. So let's take a look at our protein structure once again. So because enzymes are made of a long chain of amino acids, they're joined together by peptide bonds, and they'll have a secondary structure too with connections between different amino acids through hydrogen bonds, and then of course a tertiary structure that gives it its three-dimensional shape. And the shape is very important because it determines the active site where the reaction is going to happen with the substrate. Now, just like any protein, if the conditions are unfavorable, let's say things get too hot, those bonds can start to come apart and this enzyme can lose its shape. So it unfolds or denatures, and then we may not have an active site that's the same shape as it should be to interact with the substrate. So denaturation is really going to affect the reaction rates because the structure of the protein is no longer there to be able to fit with the substrate and for it to attach and for the reaction to occur. Now, there are other ways that enzymes' activity can be affected as well. It's not just temperature. This can be pH. This can be the amount of enzyme there, the amount of substrate there, like we already mentioned. It could also be the presence of inhibitors. So inhibitors are other molecules that can either attach at that active site and block the substrate from binding, so that's called competitive inhibition, or they can attach at other points on the enzyme at an allosteric site, is what it's called, and that might cause some configurational change in the enzyme as well and cause the active site to no longer be compatible with the substrate. Both of these will affect the enzyme's activity. So let's get back to important things to remember about enzymes. They are proteins. They are specific to the substrates that they act on. So amylase is not going to do the same thing as catalase or lactase or DNA polymerase. They do remain unchanged after reaction, so they're reusable. You can use them again and again. They work in a specific environment. So many enzymes have what's called an optimum pH or an optimum temperature where they function best. We'll see the rate of activity is going to be the highest there. And in general, enzymes work by lowering the activation energy of a reaction. Many times, biological reactions can occur without the presence of an enzyme. It just takes more energy to get there. So enzymes work by lowering that activation energy, making it easier to undergo the reaction and having more reaction activity within the cell. Photosynthesis is the process of using sunlight and carbon dioxide to make food in plants. We're going to get a little bit more specific and revise this just a little bit because we know not only plants do photosynthesis, but also algae and some bacteria. And it's not just food that they're making. So we're going to start talking about glucose, that organic compound. It's a carbohydrate, C6H12O6. So our basic equation for photosynthesis is sunlight plus carbon dioxide plus water make or yield, that's what that arrow means, glucose and oxygen. So plants are taking in these things, sunlight, carbon dioxide, and water, and then they are making glucose and oxygen during the process of photosynthesis. So if we start big at this tree and then we zoom into a leaf where the green stuff is, that's where photosynthesis is going to happen. We look on the outer layer of the leaf and we see different types of cells. These are actually the epidermal cells on the leaf. And then you might be able to see these little green dots inside these cells on the micrograph, and those are the chloroplasts. Chlorophyll is the pigment within chloroplasts that makes them green, and it's the chlorophyll that's actually absorbing the sunlight in the process of photosynthesis. So let's talk a little bit more about these ingredients. Sunlight is sunlight. Carbon dioxide can also be abbreviated as CO2, C for carbon, O for oxygen, meaning there's 2 atoms of oxygen and 1 atom of carbon in each carbon dioxide molecule. Water, of course, is H2O, and glucose is C6H12O6, and oxygen, of course, is O2. So this is our full equation for photosynthesis, not balanced. Sometimes you'll see the 6's in front of these molecules to make the balanced equation. What's really important is that the glucose and oxygen that are produced in photosynthesis are going to go directly into the process of cellular respiration, which cells use to make energy. And remember, plants don't just do photosynthesis, they also do cellular respiration. Cellular respiration takes place in the mitochondria. Photosynthesis takes place in the chloroplast, but plant cells have both. As you can see here in our very simple plant cell diagram, here's our chloroplast and here are 2 mitochondria. One thing your teacher might ask you is to reverse this equation and see what it might look like, and it definitely looks very similar to the equation of cellular respiration because the inputs of one are the outputs of the other. All right, so our definition for photosynthesis at the AP level is a little bit more complex. One way that it's stated is coordinated reaction pathways that capture energy from the sun to yield ATP and NADPH, which power the production of organic molecules. Phew. Well, we know that those organic molecules are going to be things like glucose, and we have this really important part here about how this process captures energy from the sun, and in fact, all life on Earth gets energy from the sun because of photosynthesis. Remember, photosynthesis takes place in chloroplasts, and we think that the very first chloroplasts were actually prokaryotic organisms that performed photosynthetic-like pathways, and that they were engulfed by larger single-cellular organisms who, after a very long time, formed a symbiotic relationship together and developed into the chloroplasts that we know today. This is part of the theory of endosymbiosis, and some of the evidence pieces that point to this are the double membrane that surrounds the chloroplast and the fact that the chloroplast itself has its own DNA. All right, let's get to the good stuff. There's 2 main parts of photosynthesis that we're going to study at the AP Biology level. We have the light-dependent or the light reactions of photosynthesis, and we have the dark or carbon fixation reactions. Now remember, all of this is taking place within the chloroplast, and the space in the chloroplast is called the stroma, but then there's also a third membrane, this thylakoid membrane, and these membranes are going to be able to provide us with concentration gradients for some really essential parts of the process to happen. Now, the light-dependent reactions are dependent on light, and so sunlight is going to be absorbed by chlorophyll pigments at first, and water, one of our main ingredients for photosynthesis, is going to enter here. Now we have complexes of proteins and pigments called photosystems, and these photosystems, photosystem 2 and photosystem 1, are basically there to split the water and pass electrons down an electron transport chain, very similar to the electron transport chain that we see in cellular respiration. But in this electron transport chain, the final electron acceptor is NADP+, which will yield NADPH, a very important ingredient for the next part of photosynthesis. This process also is going to generate a proton gradient, and we also have ATP synthase here, just like we have in cellular respiration. And so ATP synthase will generate ATP, and that ATP will also help with the next part of photosynthesis. So again, those light reactions take place within the thylakoid or across the membranes of the thylakoid. They involve protein complexes called photosystem 2 and photosystem 1. It is a little confusing that 2 comes first, but it was discovered second, so, scientist, what can you do? Water goes in here, water is split here, and oxygen comes out of that splitting of the water. So all of the oxygen on Earth actually comes from the light reactions of photosynthesis here. In AP Biology, you do not need to memorize all the steps in this electron transport chain or the finer details of it. These are the main components you will need to know about this part of the process. Your teacher may go into more detail about the different protein complexes or what's happening with the electrons here. But it's really important to know that at the end of the light-dependent reactions, we have ATP and NADPH to power the dark reactions or the carbon fixation reactions that happen outside the thylakoid in the stroma of the chloroplast. These reactions are part of a cycle called the Calvin cycle, and in the Calvin cycle, carbon dioxide enters, and eventually, with the help of ATP and NADPH, sugars are produced. In AP Biology, you do not need to memorize the steps of the Calvin cycle, but you do need to know what goes in and what comes out of it. Now, these sugars are not going to be glucose immediately after the Calvin cycle. In fact, it takes a couple more turns of the Calvin cycle and a few more steps to get to glucose, but you can say sugars or organic compounds are generated here. All living things need energy, and one of the main ways they get energy is through a process called cellular respiration. Often when we say cellular respiration, we're actually referring to aerobic cellular respiration, meaning the process by which we use oxygen and glucose and then undergo lots of different reactions to get ATP, the cell's energy, and we produce the byproducts, carbon dioxide and water. If we think about this on an organismal level, this is happening across our body in trillions of cells, but the glucose we get from consuming, so we take in food, we digest it, and we get glucose from the food that we eat, and oxygen we inhale, and then throughout our cells, we're going to generate carbon dioxide as a byproduct. So we exhale that excess carbon dioxide, and then we exhale water vapor as well, and that comes from cellular respiration too. Now the ATP we don't really exhale, we use that in our cells for things like active transport and other processes that require energy. But cellular respiration takes place anywhere there's mitochondria. So virtually all eukaryotic organisms that have mitochondria are going to use this type of energy pathway. Now even organisms that perform photosynthesis, like plants, will also have mitochondria because they'll take those products from photosynthesis and then they'll go into our cellular respiration reactions. Now if we look at our equation, our summary equation for cellular respiration, it's glucose and oxygen yield, water, carbon dioxide, and ATP. Again, this is the unbalanced version of that equation. And then if we look at the reverse, sort of, we see that photosynthesis looks very similar to the cellular respiration equation because, in fact, the inputs of one are the outputs of another. Virtually. We're not going to talk about sunlight too much here. So we see that in cellular respiration, we need oxygen and glucose, which is produced from photosynthesis, and then as a byproduct, carbon dioxide and water are generated in cellular respiration. The energy that's generated in cellular respiration doesn't go back into photosynthesis. It's sunlight energy that's captured for photosynthesis, and then that process can feed cellular respiration. So let's level it up a little bit and talk about these three main parts of cellular respiration. Now, if you want a cool way to review, I like this mnemonic, going crazy can offer purpose, which stands for glycolysis, Krebs cycle, and oxidative phosphorylation. Krebs cycle is also the citric acid cycle. You may have learned it either way or both ways, but I like this little mnemonic here to remember all the individual parts. Now, glycolysis is the first part. It occurs in the cytosol, so outside the mitochondria, and then we're going to be in the mitochondrial matrix where the citric acid cycle occurs. Finally, we have oxidative phosphorylation in our electron transport chain, and that's across an intermitochondrial membrane because we have to establish a proton gradient to get ATP synthase working. And remember, oxygen is our final electron acceptor at the end of all of those pathways. But remember, just overall, cellular respiration metabolizes organic molecules, so it's breaking those down for energy, and then it's returning carbon dioxide to the atmosphere. And then photosynthesis removes carbon dioxide, reduces CO2, and then fixes that carbon into our organic molecules, eventually glucose. Oxygen is truly an important ingredient, but sometimes some organisms who are capable of performing respiration cannot do aerobic respiration because they don't have enough oxygen in the cell or in the mitochondria. Other times, other organisms can only perform anaerobic processes. Now, anaerobic, an means not or without, aerobic with air, so not with air, means without air or oxygen. Now, here's where it gets a little tricky, and sometimes teachers will call this the same thing. Anaerobic respiration is technically when we go through that electron transport chain at the end of our cellular respiration reactions, and there's something other than oxygen to accept the final electrons. Fermentation is an extension of glycolysis. So after glycolysis, we've generated a little ATP, then there's going to be some more reactions that the cell undergoes without going into the other steps of cellular respiration. So we're not going to do the Krebs cycle. It can be done without the presence of mitochondria. So just be aware that aerobic cellular respiration involves oxygen and anaerobic respiration does not. Now, in comparison, one of the main things you want to remember is that compared to aerobic cellular respiration, which produces 36 ATP, fermentation only produces 2 ATP. Now I have a little bacteria on the screen because many bacteria can perform fermentation, other single cell organisms like yeast often do it, but we can also do it in our cells as well. Now there's different types of fermentation, which we'll get into. So in our muscle cells, for example, this is one type of fermentation here, which is called lactic acid fermentation based on one of the byproducts. So instead of having glucose and oxygen working together, we just have glucose. And at the end of these reactions, we are left with lactic acid and ATP. Now, we used to think that when our muscles get really sore in workouts, it's because of this lactic acid and because our body's been doing so much lactic acid fermentation. Scientists are now kind of debunking that idea and saying that the soreness is not really from lactic acid, it's more likely from little micro tears in your muscles. But lactic acid is a byproduct of lactic acid fermentation, and we do perform it in our muscle cells when we run low on oxygen. Now there's another type of fermentation, alcoholic fermentation, which can happen in yeast, it can happen in some bacteria. This process can also be completed without the presence of oxygen, and it produces carbon dioxide ethanol, which is a form of alcohol, and ATP. Still just 2 ATP, but it has those other byproducts, carbon dioxide and ethanol. Now let's take a look at this chart comparing the products and what goes in and what goes out of each of these. So within cellular respiration, remember we need glucose and oxygen. That's what makes it an aerobic process. Aerobic process, not anaerobic. And then the products are 36 ATP molecules and carbon dioxide. Now, in our two types of fermentation we just talked about, lactic acid fermentation also requires glucose. Alcoholic fermentation also requires glucose. They are both anaerobic processes, meaning they occur without the presence of oxygen. They both produce 2 ATP. Lactic acid fermentation has also lactic acid as a byproduct, and alcohol fermentation produces ethanol and CO2 as byproducts. Cell signaling pathways, or signal transduction pathways, that they're characterized by a signal, transduction, and a response. They are highly specific and regulated, so it can't just be any molecule that causes a response to happen in a cell. And many times one signal molecule can cause a cascading effect, releasing thousands of other molecules, amplifying a signal within a cell. And a lot of these pathways evolved from common ancestors millions of years ago, and they're conserved across many species. Another piece of biochemical evidence in our collection of evidence for evolution. Now, we can have unicellular signaling pathways between organisms that are just single-celled, or we can have multicellular signaling pathways that happen within multicellular organisms, sending from one part of the body, one endocrine gland, all the way to another part of the body and having some pretty large effects. Now, we have lots of different types of signals, especially within the human body. Autocrine signals go from one part of a cell to another. We can have synaptic signals, so these are going to cross synapses and neurotransmitters. We can have paracrine signals, which are released from one cell and go to another nearby cell. Endocrine signals like hormones, which can travel across the bloodstream long distances. Maybe they're released by one gland, like the pituitary gland, and then they go a very long distance in the body to have many different effects. So very simply, when a ligand, or that signal molecule, binds to a receptor protein on the membrane outside the cell, it can cause a conformational change in the receptor, which results in the activation of this signal transduction pathway within the cell. Eventually, those signals will cause something to happen, and we'll talk about what those effects could be, and they may even get to the nucleus of the cell, which is shown here. So they can get pretty complex. This is just one particular signaling pathway. If we have the activation of several different enzymes, we could have thousands of different proteins within the cell. This is why we call it an amplification response, activated and turned on. So a few I wanted to talk about here are G protein linked receptors. So this is a type of protein that's across the membrane. It can receive a signal from something like a hormone, a neurotransmitter, another chemical messenger. And then we have activation when that signaling molecule binds, which causes the receptor to change its shape. And then that activates the G protein inside the cell, which is going to go to the next molecule, sending the message along. And then that can trigger a series of events in the cell, which leads to a specific response. Now, sometimes we get that message sent to something like a protein kinase. And these are enzymes that phosphorylate, and they can add phosphate groups to other proteins. And a lot of times this activates the other proteins, so it changes the shape, adding that phosphate group that are going to target other proteins that are involved in the signaling pathway. A lot of times we'll have secondary messengers involved too. These are small water-soluble molecules that can quickly diffuse within the cell, and they'll move to signal on other parts of the process. Cyclic AMP, calcium ions, these are all really common second messengers in signal transduction pathways. Another type of important part of cell signaling that's in the cell membrane are ligand-gated ion channels. And so when they're activated, there's going to be a flow of ions across the membrane, which is going to influence the membrane potential and then have an effect on electrical signaling within the cell. Okay, so let's talk about after all these signals are happening, we have our transduction within the cell. What is going to happen when it gets to the nucleus or it gets to the location for its response? So a lot of times this is things like gene expression. It's going to turn on a particular gene. We're going to undergo transcription. We'll get to that when we get to protein synthesis later on. So we'll have some other type of protein activity. We can even trigger apoptosis, which is programmed cell death. And a lot of times this is a good thing for the health of the organism. Many cancers arise because we have inactivations of these signaling pathways, and programmed apoptosis is not occurring in cells that should be dying. So again, we can turn a gene on, we can have the activation of an enzyme, we can even trigger cell division, and we'll get into some of these specific signals a little bit more when we talk about the cell cycle and mitosis. I want to mention hormones again very quickly. These are signals. We can have steroid hormones, which are usually cholesterols, and they can pass through the cell membrane. And then we have non-steroid, which are more water soluble, and these usually are going to act with receptors on the membrane that'll cause a signaling pathway to start or interact with the receptor that allows it into the cell. So feedback is used by organisms to maintain their internal environments and respond to internal and external changes. We can also see feedback loops at the cellular level, the organismal level, or at an environmental or population level. Feedback is a response within a molecule, cell, organism, even an ecosystem that influences the continued activity or productivity of a system. Feedback loops help regulate homeostasis, or the internal balance all organisms need to survive. So the failure to maintain homeostasis can cause disease or death, meaning that the failure to maintain feedback loops can also lead to disease or death. Feedback loops are really important in biologic systems. I want to review some of the types of them right now. There are both what we call negative feedback loops and positive feedback loops. These don't mean something that are good or bad, rather they refer to the inhibition or slowing down of a system versus the amplification or the increase of a system. Negative feedback loops result in the inhibition or the slowing down of a process, moving something back to that set point, that internal balance that an organism wants to maintain. Positive feedback loops, on the other hand, amplify a signal or some sort of output. Now, negative feedback loops, like I said, will work to maintain homeostasis and go back toward a set point. Positive feedback loops are generally going away from a set point, and they're working against what the normal system would normally be. This isn't a bad thing, but generally it's not normal. In negative feedback loops, these are more common within organisms, and they maintain various body systems. Our temperature regulation, our blood pressure regulation, our blood sugar regulation are all examples of how negative feedback loops help us maintain our internal environments. You may see an example of blood pressure or blood sugar regulation on an AP exam, so we're going to go over what that could look like in just a moment. Positive feedback loops, though less common, are still necessary in certain situations. Childbirth and having the contractions of the uterus in order to deliver a child is obviously really important for mammals. Fruit ripening, producing ethylene, for example, is another positive feedback loop that's necessary for the ripening of apples on a tree. Lactation in mammals also is helped by positive feedback loops and the release of oxytocin, and blood clotting as well, which we'll go over as an example in this video. So a negative feedback loop, remember, is going back towards a set point. So if we start with a normal blood pressure and then an organism is exposed to stress, the blood pressure would rise above normal. Now the body receptors are going to detect change and then decrease a heart rate and increase the blood vessel diameter. These effects will then help decrease the blood pressure and bring the blood pressure back to normal, back to that set point. This feedback loop would then stop here unless the body is exposed to more stress. A positive feedback loop related to blood clotting kind of works in a different way. If we have a break in a blood vessel wall from a cut or a laceration, platelets are then going to adhere to the site and start to release chemical signals. Those chemicals will then attract more platelets, and platelets will adhere to the site and release more chemical signals. This will continue to amplify the signal, which doesn't normally happen in the body, and more platelets will come until finally we have what's called a platelet plug that is formed at the site of the break. Then the cycle ends, we don't have any more chemicals to attract more platelets because we don't need them anymore. Similar with childbirth. Once the child is delivered, the contractions don't need to amplify because the goal has been achieved. The DNA in a chromosome isn't actually just one strand with, a couple dozen bases. In fact, it is millions of bases. Many chromosomes contain 50 million nucleotide base pairs, some even 300 million nucleotide base pairs. So what we're seeing here is a very bad representation of a chromosome. Remember, chromosomes are DNA that are tightly condensed and packed, wound up around specific proteins called histones. So let's talk a little bit about that organization and then we'll get into the vocab. So if we have our DNA molecule, we know that in the middle it's connected with base pairs, there's a backbone. When it starts to be wound up, it is wound around these protein complexes that can unwind to be transcribed or expressed. And when it prepares for cell division, it has to be very tightly wound into these structures called chromosomes. So that winding of the DNA around these proteins is going to be very important for us to organize the DNA in a way that allows it to be distributed evenly into the daughter cells. So when it is in the nucleus and it's not going through mitosis, it's not wound compactly into chromosome forms, we call it chromatin. Chromatin is just that complex of DNA and protein. Sometimes it's shown here, you can see it kind of tiny, it's sort of this like spaghetti-like substance. Now, when it prepares for cell division, the DNA, and you can kind of see it here, will condense into those chromosomes. In humans, we have 46, so it'll condense into those 46 individual chromosomes. Remember, these are long, long strands of DNA that are being packed tightly together. And before it goes through mitosis as well, the DNA has to duplicate. So each chromosome actually has 2 identical sister chromatids. They're the same on each side. And in mitosis, those chromatids are going to split apart. So let's talk about that vocab one more time. This whole thing is a chromosome, and you can see the individual chromosomes in the nucleus, but when it's not condensed into the chromosomes, we call it chromatin, or just that bundle of DNA and proteins is chromatin. The chromosome is attached at the center by a centromere, so centromere, center. And then each side of the chromosome has an identical sister chromatid. So one section here, that's one chromatid, and one section here, that's one chromatid. So one more time, chromosome, chromatid, centromere, and chromatin here in the center. Okay, we're going to be referencing these throughout today's reviews. I just want to make sure you had them all straight. We want to talk about the cell cycle, which is how a typical cell is going to go through its life. And so the main part of a cell's life cycle is called interphase, which involves stages G1, S, and G2, and that takes up the bulk of the cell's activities. So this interphase stage actually lasts a really long time. G1 is for growth, S is for synthesis of the DNA, so where the DNA is duplicated, and G2 is also for growth and preparation for division. After the cell divides, it can continue going through this cycle again and again, as long as the right signals are being processed within the cell. If we zoom in and talk about mitosis and cytokinesis specifically, we're going to look at the main stages of mitosis for how the cell is going to divide its genetic information so that its daughter cells have the exact same stuff as the parent cell. We can remember the stages of mitosis as PMAT, prophase, metaphase, anaphase, and telophase. And if we look on here, prophase involves the DNA condensing, our nuclear membrane dissolving, and our chromosomes sort of migrating to the center of the cell. Metaphase is going to involve those chromosomes lining up in the middle of the cell along the cellular equator, and the spindle fibers are going to start to attach to the centromeres of the chromosomes. In anaphase, those sister chromatids are pulled away, A for away, from each other, and those spindle fibers are going to pull them to opposite ends of the cell. In telophase, we start to get new nuclear membranes reforming, and then in cytokinesis, the cytoplasm is actually going to split, and we're going to have a cleavage, and we're going to get 2 daughter cells. The DNA is decondensing and going back into chromatin. All right, so again, a couple of things to remember about mitosis. In a regular body cell, we're typically going to go from a diploid cell with just the regular amount of chromosomes, which is represented by 2N, to 2 daughter cells that are also diploid. So in mitosis, we go from diploid to diploid. We go from 1 cell to 2 cells, and they have identical genetic information as the parent cell. So the point of this is to make 2 identical daughters. The DNA has to be duplicated in S phase before it can undergo mitosis, or else if we split the DNA in half without duplication, we would end up with half the DNA in the daughter cells, which is not what we want in the process of mitosis. That's for meiosis, which we'll talk about later on. So S phase has to happen during interphase, this longer stage of the cell cycle, in order for the DNA to be duplicated, and we can have enough in order to split it later down the road. Okay, so again, once we've duplicated that DNA in prophase, it's going to condense and get ready for cell division. Metaphase line up in the middle, anaphase pull apart, telophase, we're going to get 2 new nuclear membranes starting to form, and then cytokinesis, we're going to get cleavage, and we're going to cut those cells in half. Okay, let's talk about the cell cycle now and these checkpoints that are regulated throughout a cell's life cycle. The cell cycle has multiple checkpoints or control points where we have these stop and go signals that are going to regulate how the cell cycle will continue. A lot of this is dependent on proteins and things called cyclins. And these cyclins are proteins that are going to go up and down in different concentrations in the cell depending on which stage of the cell cycle it's in. They're going to regulate various stages of the cell cycle through signal transduction division pathways by binding and activating different proteins called cyclin-dependent kinases, which have the ability to phosphorylate or add phosphate groups to other target proteins. So we have different cyclins and different cyclin-dependent kinase complexes that are going to regulate different stages of the cell cycle. And so they're going to go up and down at various checkpoints. Just a few of some of the important checkpoints in the cell cycle are our G1 checkpoint, which will allow it to go through S phase, DNA replication, G2, and then mitosis and divide. But if it doesn't get the go-ahead at that checkpoint, then it can enter a G0 phase, which is a non-dividing state that a lot of body cells end up in. Another one here, at the M phase, is going to ensure that all the spindle fibers are properly attached to the centromeres of the chromosomes and they're all lined up in the right way. And if the cell passes that checkpoint, it receives the go-ahead signal, then it allows the cell to proceed into anaphase, separating those sister chromatids and pulling them to opposite ends of the cell. But if some of the fibers are not attached to the chromosomes in the right way, then the cell is going to receive a stop signal and it should not continue dividing. Let's take a look at another one, the G2 checkpoint. So we have the activation of MPF or mitosis promoting factor here. We're going to have a gradual rise of cyclin levels during G2 here, and then cyclin B can bind to CDK1, and then our MPF complex becomes active, which triggers the onset of mitosis. And this only is going to occur after the proper checks within the cell ensure that DNA replication is complete and that the cell is entirely ready for cell division. Now, cancer eludes a lot of these checkpoints. So if we have any signaling pathways that are not functioning properly within the cell, especially related to some of these checkpoints, that's when the cell is going to divide when it shouldn't. We could have unequal distribution of chromosomes. We could have cells that ignore signals for apoptosis or programmed cell death, or ignoring that checkpoint during G1 to enter a non-dividing state. So remember that cancer is a loss of control of these checkpoints throughout the cell cycle. And usually that's because there's a malfunction in a gene that controls the proteins involved in these signaling pathways. A lot of cancer cells will continue dividing indefinitely in the lab if they're given all the nutrients they need, because they can just bypass the checkpoints whenever they want and continue to divide and divide. The phases of mitosis can be remembered as PMAT, or prophase, metaphase, anaphase, and telophase. And with meiosis, you might have heard that we go through PMAT twice, which is true, but we are going to add in some additional information today as we're talking about our review. Now, one of the main differences between meiosis and mitosis is the end goal. Mitosis's goal is to create two identical daughter cells. Meiosis, the end product, is 4 haploid daughter cells. And haploid means it has half the genetic information of the parent cell. Parent cell starts out as diploid, and then each of those daughter cells have half of the original information. This is useful for creating gametes or sex cells so that they can combine in fertilization to then create new diploid cells. One way I like to remember the difference so I don't get mixed up is thinking about mitosis is useful for growth and development and repair in larger multicellular organisms. So if I got a bump or a scratch on my toe, I would go through mitosis. In order to fix that, my cells would divide and create exact copies. But to generate me, I had to come from an egg cell and a sperm cell, and each of those was generated through meiosis. So me, meiosis, my toe, mitosis. All right, so here are all the stages of mitosis together. We have prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis, and then we do it all again. Prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis. So let's talk about what happens in each of these at a little bit closer level. So in meiosis 1, we have prophase 1 first, and our chromosomes condense and become visible, just like they do in prophase of mitosis. Our homologous chromosomes are going to pair up, and we're going to have a process called crossing over, where we have an exchange of genetic information between homologous chromosomes. This is really key in increasing genetic variation in organisms, but we have chromosome from one parent, chromosome from the other parent, and they're lining up together and they're exchanging genetic information through a synapsis. And after this crossing over occurs, we have a pretty unique combination of genes from both parents, and this is why no two siblings, unless they're monozygotic or identical twins, are going to have the exact same DNA. In metaphase 1, we're going to have our homologous chromosome pairs lining up at the cellular equator. And then instead of separating the sister chromatids, in anaphase 1 of meiosis, we have homologous chromosomes separated by those spindle fibers. And so at the end, we end up having full duplicated chromosomes still at the end of telophase 1. Then our cell splits, then it's time to do it all again. Remember, at this point, sister chromatids are still attached. We have to separate them in the next part of meiosis. So in meiosis 2, we have 2 rows here because we have 2 cells already, and remember we're going to end up with 4 cells, so we have to undergo this division again. In prophase 2, our chromosomes will prepare again for this division, condensing if necessary. Our spindle fibers are going to form, and then in metaphase 2 we have our chromosomes lining up at the cell's equator. Notice that this part of meiosis looks a lot like mitosis. This part is more closely aligned to what we see in mitosis. So we have those chromosomes lining up just in one row this time on the cellular equator, and then in anaphase 2, sister chromatids are being pulled apart to opposite ends of the cell, and then in telophase 2, we have our genetic information fully separated, a nuclear envelope starts to form again, and then we're going to have cytokinesis when that actual split occurs, resulting in 4 daughter cells with half the genetic information of the original parent and different combinations of those genes as well. And here's the full process again, but I invite you to look in your textbook or any other diagrams your teachers provided to get a closer picture. Species have to be able to reproduce in order to pass on their traits and their genes and to survive. Some organisms reproduce asexually, and virtually all of their genes come from the same parent, and so they're copies of the parent organism. Other organisms reproduce sexually and have half of the genetic information come from 2 different parents. So one half comes from one parent and one half of the genetic information comes from another parent, and those 2 haploid cells are combined. Remember, in order to get those haploid cells or gametes or sex cells, for sexual reproduction, we have to go through meiosis first. The process of meiosis results in half the genetic information in the daughter cells, and in humans, this happens during the production of eggs or sperm. We can get lots of genetic variation through meiosis, through independent assortment of chromosomes, crossing over of chromosomes during prophase 1 of meiosis, and then of course the random fertilization of one egg cell with any one sperm cell. Remember, mutation is an important source of genetic variation, but these 3 reasons on this slide are very, very important as well. The process of meiosis is important in sexual reproduction because it provides genetic variation of offspring, and genetic diversity is important for the survival of a species. So in order to get that egg and that sperm, we have to go through the process of meiosis first. Then those 2 can undergo fertilization to produce a zygote, which is a fertilized egg cell and now a diploid cell. So let's put this all together. In humans, both a biological male and a biological female will need to undergo meiosis in order to produce eggs and sperm. Then one egg cell and one sperm cell will combine in fertilization. After that, that fertilized egg will divide and make copies of itself through mitosis as the embryo starts to grow. Mitosis will continue until the cells start to differentiate and become all the different important parts of the growing embryo. Right, and if you have to memorize Mendel's laws, an easy way to remember them is pretend that at the monastery, Gregor Mendel, who was a monk, had a nickname named Sid. I don't know why, it's just part of the story, but Sid will help you remember the 3 main laws that he came up with: segregation, independent assortment, and dominance. A gene or genes are pieces of DNA that determine your traits. An allele is just a different form of a gene. If we're writing out genetics problems, this might be written as a big B or a little b, for example. A recessive gene can be masked by a dominant one, and a dominant allele can mask a recessive one. Often dominant alleles are represented by capital letters. A genotype is the actual combination of alleles that somebody has. A phenotype are the physical characteristics that come from that genotype. So for example, your physical characteristic could be tall. That's your phenotype. Homozygous is when we have an allele pair that has 2 of the same allele. So for example, a little a, or a big A, big A. As many human traits are caused by combinations of up to thousands of different genes interacting together. And the interactions of different genes can cause differences in phenotypes, and we have a spectrum or a range of different phenotypes in many instances. For example, hair color, skin color, it's not just a yellow pea or a green pea. It's much more complex than that. And then of course we have epigenetics too, where we have different factors that are controlling which genes are turned on and off in which environment, so it does get very complex. But today what we're going to talk about are patterns of inheritance where we don't just have one single dominant allele and one single recessive allele. We can have things such as incomplete dominance. This is when we have a blending of traits. So there may be 2 different alleles here. For example, the classic one is something like a red flower and a white flower. We'll pretend this is white on the screen. And then if they are crossed or bred, they would generate pink flowers. So let's see how this works. So let's say that in this incomplete dominance scenario, we're going to represent red flowers with big R, white flowers with big W, big W. Notice how these are 2 capital letters for both alleles. And we're using different letters too instead of R's. That would get really confusing if they were both big R's here. So we set the Punnett square cross up like we would do any classic Punnett square, and we see that our combinations in this case could only be big R, big W, RW, 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 RW. And so what that results in is a phenotype that's a blend of the two parent traits. So RW would actually give us pink, that intermediate characteristic, instead of one or the other, which is really interesting. Codominance is when we have a representation of both traits in the organism. So we're not meeting in the middle, but instead we have both traits show up at some level. Roan cattle are a very common example of this. But with flowers, again, to do it very simply, let's say we have purple flowers and pink flowers. Instead of getting a blend of that purple and pink, we would get some petals that are purple and some petals that are pink, or some spots in the petals that are completely purple and some spots in the petals that are completely pink. So let's see how this works with the Punnett square. In this case too, we use capital letters, two different letters to represent the two different traits. Big P for purple, big B for pink. We cross these two. What we end up getting is, just like we saw in the previous example, these heterozygous offspring that are a combination of both the purple allele and the pink allele. And so what that gives us are flowers that are not a blend of the two colors, but instead show both traits. Blood types we'll see as an example for codominance very frequently. Now, blood types are very interesting. They are controlled by several different alleles. So we have something called multiple alleles that are a possibility. It's not just A or B. We can have the allele for blood type A, the allele for blood type B, or the allele for blood type O. So that's 3 different alleles. And then remember, a genotype is 2 alleles together. So we have lots of different possible combinations here. And AB blood is actually a codominant genotype. So if someone has the allele for type A and the allele for type B, they will represent antigens for both blood type A and blood type B. And then we can talk about really interesting patterns that occur because type O is actually recessive, but A and B are codominant. So that's a good example of codominance. And then of course there's the Rh factor and lots of different things we can talk about with the different blood types and who's a donor and whatnot. But let's move ahead now to talk about sex-linked traits, which is another different type of inheritance that we see in humans. So these are traits that have alleles that are located on sex chromosomes. So in a human karyotype, there are 23 pairs, so 46 total chromosomes, and 2 of those are sex chromosomes. In biologic females, it's an X and an X. In biological males, it's an X and a Y. If something is an X-linked trait, that means it appears on the X chromosome. Generally, there's going to be more X-linked traits that we'll talk about just because the X chromosome is much bigger and there's more space for traits. Colorblindness is a very common X-linked recessive trait, as is hemophilia. That is also X-linked recessive. And Y-linked traits, there's fewer of them that we'll study and talk about just because the Y chromosome is smaller. And many Y-linked traits actually lead to sterility. So if someone had one of those Y-linked traits, they could not pass it on to their offspring because if someone is sterile, they cannot have offspring. All right, so let's just take a look really quick. Remember, females have the XX chromosome pair and males have an X and a Y. If we look at a human karyotype, so 1 through 22, these are all the autosomes or the non-sex chromosomes that humans have, and then the 2 sex chromosomes are either XX or XY, and so this is a karyotype of a human male. So let's take a look at the inheritance for color blindness. So the allele for normal vision is dominant, and the allele for color blindness, or someone who is affected with this color blindness trait, is recessive. So we'll show that with a little h. Here we show 2 normal individuals. Neither of them carry any of the genes for color blindness. In this case, this female is a carrier here. She is still normal though because she has one dominant allele, that big H, and that can mask or cover up the little h. So she is still fine. She has normal vision. She is not colorblind. The male here is normal as well. Now in this case, the female is colorblind. She has 2 of those alleles for colorblindness. They're both carried on the X chromosome. The male is also normal. And here we have 2 colorblind individuals. We have a colorblind female and a colorblind male. But notice with the male, he only needs 1 copy of that recessive allele in order to be colorblind because he only has 1 X chromosome. So there's no dominant allele present to be able to mask that other allele. And so he will be colorblind with that 1 copy of the affected allele. And here we have a case with a female who is normal. Her alleles are unaffected and the male is colorblind. All right, so how do we set up a Punnett square for a sex-linked cross? It's not overly complicated. We just have to make sure we carry the X's and the H's or whatever letter we're using for the trait together. We don't separate those, but we are going to separate each individual sex chromosome. So if we were to cross these two parents, we would set up the Punnett square like this, and we make sure we write the letters as like an exponent, like it shows in their genotypes up at the top. And then just like a regular Punnett square, we'll carry down the letters, bring them across to see the possible combinations for the offspring. So in this case, we could have, out of all the possible offspring, there's a 50% chance that this couple will produce females who are carriers for color blindness but not have color blindness themselves, and a 50% chance that they will have offspring who are males who are normal, they don't carry any of the colorblind alleles. So remember, females can be carriers of sex-linked traits because they have two X chromosomes, and technically in biology a carrier is someone who does not display the trait but they could pass it on to their offspring. So in this case, a carrier for color blindness would look like this, and males technically cannot be carriers of sex-linked traits. So obviously someone who has the trait could pass it on to their offspring, but a carrier, if we're going by that strict definition, males can have conditions that are sex-linked, but they're not carriers technically. Genes can be influenced by the environment. For example, in identical twin studies, we can see that identical twins separated at birth can have different heights and weights after 20 years. Most likely the cause of these differences is that each twin was provided a different diet and had different physical activities throughout their lifetimes, because an environment does play a role in influencing individuals. So let's look at a karyotype. Now remember, a karyotype is just an image of all the chromosomes in the nucleus of an organism's any one cell. So in humans, we have 46 total chromosomes, or 23 pairs. So remember that the first 22 pairs, these are called autosomes, and the two that are left, these are our sex chromosomes. If you're biologically female, you have two X sex chromosomes. If you're biologically male, your sex chromosomes are X and Y. Now we do see some differences in certain genetic disorders where we can have different combinations of these, but we'll get into that when we start talking about genetic differences. So how do we get a karyotype of a baby that's not born yet? Well, we can use something called amniocentesis. Amniocentesis, or an amniotic fluid test, is a medical procedure used in prenatal diagnosis of genetic disorders and fetal abnormalities. So what happens is a small amount of amniotic fluid, which is here, is going to contain some fetal tissues, and it's going to be extracted from the amniotic sac with a large needle. And once we take it out, the DNA can be examined for genetic abnormalities, or we can simply see the biological sex of the individual. Now, not all women choose to do this because there is a small risk involved in the procedure of amniocentesis, but if you elect to do it, or if you're high-risk pregnancy, some women will be able to see a karyotype of their child before they're born. Now remember, a normal human karyotype has 23 pairs of chromosomes, including the sex chromosomes here. This individual has all 23 pairs, one X and one Y. If you look closely, you can see this individual is a biological male. So when we look at our karyotype, we're going to identify and evaluate the size and the shape and the number of chromosomes in our sample body of cells. Some problems that we could see would be duplicates of chromosomes where there shouldn't be, or deletions of certain arms of chromosomes, or translocations of chromosomes. One difference we could see right away is if there was a third chromosome at the 21st position. Now, all the chromosomes aren't arranged like this. Scientists have to make sure that they are arranged after the picture is taken into the correct order for the correct chromosomes. But once they are arranged, we can look and determine if there's any genetic abnormalities. For example, at the 21st location, if there's a third chromosome, we would have what's called trisomy 21, also known as Down syndrome. Trisomy just means 3 copies of a chromosome. Now, chromosomes aren't everything and genes aren't everything. The environment has a huge influence on phenotype or on the resulting traits of an individual. Some scientists estimate up to 75% of who you are and how your traits operate is due to environmental factors. We're learning so much more about epigenetics and how different things can turn genes on and off in different ways. So environmental factors can influence gene expression and lead to phenotypic plasticity. This is when individuals with the same genotype have differences in the phenotype that they express because of environmental conditions. For example, there's some mammals like rabbits that can exhibit different fur colors and different temperatures. Hydrangeas are another popular example of this. The petals of certain hydrangeas can change color depending on the pH of the soil that they're planted in. So, for example, here we see that blue flowers are going to appear when the soil is highly acidic, and pink flowers are going to appear when there's a neutral or slightly acidic soil. Many reptiles, such as crocodiles and turtles, have sex determination based on temperature because sex determination in these animals is highly dependent on hormones, and these hormones can be changed by the activity levels of different enzymes depending on a specific temperature. So we can see an entire brood of turtles hatching to be one sex because they were all incubated at a particular temperature. We know that some genetic differences in individuals are due to single allele or single gene changes, and others are due to changes in entire chromosomes. A lot of times this happens because of non-disjunction or when a chromosome doesn't separate properly during meiosis. A lot of genetic disorders we call autosomal because they're occurring on any chromosome that's not a sex chromosome. So all the chromosomes except for sex chromosomes, remember, are autosomes. And so autosomal conditions are caused by mutations on chromosomes that are not sex chromosomes. You might have heard of Huntington's disease as an example of an autosomal condition or an autosomal dominant genetic disorder, which often doesn't appear until later in life, so people can pass it on to their offspring without even knowing that they have it. And then there's lots of autosomal recessive disorders like Tay-Sachs disease, cystic fibrosis, PKU, sickle cell anemia. Chromosomal conditions are usually caused by non-disjunction, which means when those chromosomes are supposed to separate, especially during anaphase 2 of meiosis, we're getting an extra chromosome ending up in the sex cell. If you've made it all the way to the end, I wanted to share that I have more videos on my YouTube channel that dive deeper into a lot of these topics. They also have practice questions, which are a great way to study. So you can open up whichever video you need a little bit more help on and go to the section that you feel like will be most beneficial to you and look at the practice questions that I provide. I also have lots of study resources that will hopefully help you prepare for your big exams, including a 3-day study plan, how to cram for an exam in 24 hours, how to raise your grade at the last minute, all of the great stuff that you'll need to succeed. Thanks so much for watching. Give this video a like if it's been helpful, and I'll see you later.